Hello, and welcome to the recovery training for Molnupiravir. This slide shows the current recovery treatment comparisons for adults hospitalised with COVID, excluding the comparisons for flu. Molnupiravir is one of two antivirals being introduced in December 2021, and is a small molecule developed as a treatment for coronavirus infections before the current pandemic. SARS-CoV-2 uses an enzyme called RNA-dependent RNA polymerase, or RDRP, to make a copy of its genome, and this is the target of Molnupiravir. Molnupiravir is a prodrug which is metabolized to N-hydroxycytosine, or NHC, which is very similar to one of the four nucleoside letters that make up RNA. The SARS-CoV-2 polymerase incorporates NHC into the viral genome, leading to copying errors that mean that replication fails. Molnupiravir is active against a range of coronaviruses, and the barrier to developing resistance appears high, which should hopefully mean that it retains activity against future variants. In the MOVE-OUT trial of outpatients with COVID, Molnupiravir reduced the risk of hospitalisation or death by around 30%, from 10% to 7%. The MOVE-IN trial of inpatients was abandoned for futility, but only included around 150 participants on full-dose Molnupiravir or placebo. Mortality remains high in patients admitted to hospital with COVID, and MOVE-IN was much too small to exclude worthwhile benefits of Molnupiravir. We've seen previous false negative results in small trials of tocilizumab and neutralizing monoclonals, both of which later turned out to reduce the risk of death, so a larger trial is needed to identify or rule out any benefit of Molnupiravir. The Molnupiravir comparison and recovery is open to adults aged 18 and over, but some animal studies have shown fetal toxicity, so this comparison excludes pregnant or breastfeeding women, and women of childbearing potential must have a negative pregnancy test to join. Patients who've received Molnupiravir during the current illness are also excluded. There are no exclusions around liver or renal function or need for dose adjustment. The capsules are large and can't currently be opened or crushed, so patients must be able to swallow to join this comparison. The dose is 800 mg twice daily for 5 days, and unlike in previous recovery COVID comparisons, the course should be completed at home if participants are discharged before it's finished. Because time to discharge is a trial outcome, and there is no placebo control group, please try to ensure that the provision of the drug to take home does not delay discharge, as this could bias the results for this outcome. If possible, dispensing the whole course to be kept on the ward with the patient may minimise this risk. We know that the response to neutralising monoclonal antibody treatment depends on serostatus, with the benefit much greater in those who have not yet developed a specific immune response to the virus. And the MOVE-OUT trial suggested that the same may also be true of Molnupiravir. We think that biological sampling is likely to be crucial to identifying any benefit of Molnupiravir, and may be allowing us to direct future treatment in the way that we've done with monoclonals. Two different sample types are being collected in the antiviral comparisons and recovery. Firstly, a serum sample that was done with the convalescent plasmid and Molnupiravir comparisons before, and this will be used to look for antibodies to SARS-CoV-2, and also possibly to look for viral antigen. And secondly, nose swabs, which will be used to measure viral load and look at the effect of antiviral drugs on the rate of viral clearance. These will also be used to look for mutations that might confer resistance to the antiviral drugs. At each time point, a single swab will be used to swab both nostrils. Both samples will be taken at baseline, which is day 1, and these must be taken after consent, but before randomization. And swabs will also be taken at day 3 and day 5. It's important that these samples are taken not only from people allocated the antiviral treatments, but also those allocated usual care in the antiviral comparisons. Sites will be provided with sample kits for serum and nose swabs, and these will include a returned addressed envelope so they can be returned in the hospital internal mail. Full instructions for taking samples are in each kit and can also be found on the recovery website on the site Teams page. In MOVE-OUT, the rate of adverse events was similar with treatment and placebo, and the commonest reported side effects were nausea, diarrhea and headache, although these were reported in only a few % of patients, and it's not clear that the rate differs from placebo. Thank you for listening.